muscles. I'm going to pick up kind of where we left off. I'm going to review a bit just so we are on the same page here. We're going to look at skeletal muscle contraction today. So we'll look at skeletal muscle cell contraction. Skeletal muscle cells are also known as skeletal muscle fibers because they're so long and skinny. Here is a drawing of a skeletal muscle fiber up here. The sarcolemma has been peeled back. We can see a nucleus in here. Of course, there are many. We can see mitochondria, the little orange guys here. And all of these cylindrical things are myofibrils. Myofibrils are where contraction is going to take place. Here is one myofibril extending out, and we're zooming in on it down here. The two most important proteins for us in a myofibril are actin and myosin. And we actually can see them, they're depicted here at least, in this myofibril. This reddish line here going horizontally is called the thick filament. It is made out of myosin. These blue horizontal lines are called the thin filament. They are made out of actin. Notice how they overlap. We can also see this diagonal, more vertical band of proteins called the Z disc, which anchors the thin filaments. Plus, we have this more vertical line of proteins called the M line, which anchors the thick filaments. Now, in the region where we just have thin, that is called the I band. From one edge of a thick filament to the other edge, that is called an A band. And from one Z disc to the next, that is called the sarcomere. So every myofibril is going to have a ton of sarcomeres. Like one sarcomere would be from here to here, another one from here to here, and to here. So, I mean, right here I can see 1, 2, 3, 4, 5, 6, 7, 8, 9 sarcomeres. And there would be sarcomeres all the way down, sarcomeres in every single one of these myofibrils. Now, what can a sarcomere do? A sarcomere can contract. If this one sarcomere is contracting, then all the others in this myofibril are contracting, and every single sarcomere in every myofibril in this cell is contracting, and the whole cell contracts. So if we can see how one sarcomere contracts, we're going to know how the whole cell contracts. That's our plan, at least. In this depiction of a skeletal muscle fiber, still got our sarcolemma, got a nucleus right here, still see some mitochondria. I see a myofibril right here coming out, and some other myofibrils as well. The myofibrils have thick and thin filaments. They'll be arranged into sarcomeres as well. What this picture is showing me, that the other one didn't, are these pathways, these tubes through the cell called T-tubules. These are going to be important later on. Also, I can see this blue guy here. There's a blue guy basically wrapped around each myofibril. And the blue guys, each of them is called a sarcoplasmic reticulum. And the one job of theirs that we're truly interested in is the fact that they store calcium. Okay. Zooming in on another sarcomere here. I got my blue thin filament made of actin, my more pinkish reddish thick filament made of myosin. I see my anchoring Z disc. I see my anchoring M line sitting in the middle of what's called the H zone. Didn't mention that in the other slide, but we did it in the other lecture. That's okay. Now, from one Z disc to the next is a sarcomere. And what we got to figure out is how this sarcomere is going to contract. And to do that, what I want you to notice is these little roundish things. These are called myosin heads. And what they have the capacity to do is they can reach and grab on to this blue thin filament, and they can adjust the amount of overlap. Like, just think about this for a second. Let's say that these red guys grabbed onto this thin filament here. These red guys grabbed onto this thin filament. And these guys grabbed onto this one, like so. And then after they did that, they yanked this thin filament in this direction. So they yanked it in this direction. If this happened, the Z-disc would go in this direction. The Z-disc would go like this, and the sarcomere would get shorter. In other words, the sarcomere would contract. All right, let's look at some more pictures of this. Okay, before we go there, notice that if we slice through the I band, we get nothing but thin. Slice through the H zone, nothing but thick filaments. The outer edge of the A band is where I have that overlap. And that overlap is key to do what we just said. The overlap was key for the myosin to be able to grab onto the actin and then pull it inward, thus bringing the Z discs closer together and contracting the sarcomere. All right, here is a relaxed sarcomere of a muscle fiber. It's basically what we looked at. This is a depiction. This is an actual electron micrograph. So a picture of the real thing. All right, so from right here all the way to right here, that is my Z-disc to Z-disc. That's my sarcomere. So Z-disc, Z-disc. From here to here is a sarcomere. Here's my I band right here. Here's my A band. Let me actually write these down. Tell you what, let's write these down. So here's my Z, here's my Z. So from that Z to that Z is one sarcomere, okay? This line down here is my A band. This line over here is an I band, and this little line right here is my H zone. All right. So what we're going to see happen is these myosin heads are going to reach up and grab on to these actins. These myosins are going to reach up and grab onto the actins. And it's going to happen on both sides. It's going to happen on both sides. And all, just trying to show you some of them doing that grabbing. And when this occurs, then what's going to happen is the myosin's going to pull the actin in this direction. Now, when this happens, the sarcomere is going to get shorter. The sarcomere is going to contract. What do you think is going to happen to this H zone? Is it going to get smaller, bigger, stay the same? I band, what do you think is going to happen? A band, what do you think is going to happen? Sarcomere size, what do you think is going to happen? All right, let's look at a picture and let's find out. So this was the relaxed fiber. This is one that is now contracted. Look at how much more overlap we have. The zone of overlap. The zone of overlap got there we go. The zone of overlap got bigger. The zoo got bigger. The I band. The I band is tiny now. The I band got smaller. The H zone disappeared. The H zone got smaller. It's no longer even there. The sarcomere got smaller. The A band's the only thing that didn't change, because the A band is from here to here, and we didn't change the length of that thick filament. But by reaching up and grabbing onto the thin filament and pulling it inward, the sarcomere got shorter. It contracted. And if one sarcomere contracts, every sarcomere in a muscle fiber is going to contract. Every muscle cell has an all or none phenomenon. Either it contracts fully or not at all. All right. So that is how muscle contraction works. Thick filaments grab onto thin and pull them inward, shortening each of these units of the muscle cell and shortening the muscle cell itself. Okay. What we got to do now is figure out how we can stimulate a muscle fiber with a neuron and get it to contract. So here I have my muscle fiber, beautiful nuclei, nice striations, light I bands, dark A bands, over and over again. There's my myofibrils. Here's my motor end plate. Look at the waviness from our surface area, like we saw in lab. Here's my Schwann cell wrapping around the axon. Telodendrion, 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 and then here are some axon terminals. So we're zooming in on a single neuromuscular junction right there with that block. And here we go. When an action potential gets to the axon terminal of a motor neuron, of course, that action potential is going to cause calcium to rush in to the axon terminal. It's going to cause these vesicles to undergo exocytosis. And these little green guys here, the little green guys are acetylcholine. They're released into that synaptic cleft. The acetylcholine is going to bind to this receptor. By the way, this receptor right here, there's two of them that we can see. That guy right there, since he's binding acetylcholine, he is going to be cholinergic. Now, what type of cholinergic receptor is he? He is a nicotinic receptor. So that's a cholinergic nicotinic receptor. When acetylcholine binds to him, sodium is going to rush in. Sodium rushes in, and we depolarize the motor end plate. We create what's called an end plate potential, which is basically a graded potential. The graded potential moves along the sarcolemma away from the motor end plate, and on the sarcolemma, I am going to find voltage-gated channels. These two channels here are both voltage-gated. And when they're depolarized, we're going to get an action potential. And this action potential is then going to speed along the entire sarcolemma. So, neuron has an action potential. Neuron releases acetylcholine. Acetylcholine binds to the muscle cell, causes a graded potential, which then results in the muscle cell itself having an action potential. And that action potential travels along the sarcolemma. Okay. Well, what does the action potential do? Where does it go? What happens? One more thing before we get there. The acetylcholine doesn't last long. It's very quickly degraded by this enzyme called acetylcholinesterase. So we're stimulating the muscle cell, but then we're stopping. Maybe we stimulate it again. We can do it over and over again, but each stimulation is very short because the acetylcholine is quickly broken down by this enzyme called acetylcholinesterase. Okay, now what does that action potential do? We don't need to look at a graph of an action potential. I guarantee to you I am not going to have this on your test. All right? So don't sweat this. Come on. Come on. There we go. Finally, we get to where I wanted to go here. I'm just full of anticipation today. The action potential travels along the sarcolemma, and it goes down into the T-tubules. And what this does is it puts the action potential adjacent to the sarcoplasmic reticulum, which is then adjacent to the myofibril. And the T-tubule is right next to a pair of sarcoplasmic reticuli to their widened ends. Each of these widened ends is called a terminal cisterna. And this is where we are going to store a very important metal, calcium. So, the action potential goes down the T-tubule and is adjacent to these terminal cisternae. The three of them together are known as a triad, by the way, because there's three of them. All right. Action potential goes down the T-tubule. Can you guess what the action potential is going to cause the sarcoplasmic reticulum to do with this calcium that it's storing? Well, I mean, seriously, if you're storing something, what are you eventually going to do with something that's stored? You're going to use it. You're going to release it. And that is exactly what's going to happen. The action potential goes along down the T-tube. it causes a series of proteins to change their shape, which lets calcium get released. And calcium is now in the sarcoplasm. So the action potential down the T-tubule makes the cisternae release the calcium into the sarcoplasm. Okay, now what? What is that calcium going to do? Well, we can probably guess somehow that calcium is going to cause contraction. Now, let's pause for a second here. Let's think about something. We know for contraction to occur, the myosin is going to grab onto the actin and pull it inward. Well, calcium must facilitate that somehow. Let's see how. In order to see how, let's take a little closer look at that thick filament and the myosin molecules that make it up. So here is the thick filament. This is a myosin protein right here. So it's like two golf clubs kind of like twisted together. It's got a tail. It's got a bendy region. It's got heads as well. The heads have a place to bind ATP for energy and a place to grab onto actin, because we know myosin grabs onto actin, pulls it, and that's how we get contraction. All right, that's the thick filament with myosin. Here's our thin filament with actin. So here's some individual actin molecules. They got a spot for myosin to grab onto. And the actin molecules are strung together to make the thin filament. And we're going to see some proteins here. There's this stringy protein called tropomyosin. Notice that the tropomyosin is covering that little indent. And an indent was the place where myosin attached. So this is what actin and the thin filament normally looks like. And so under normal situations, myosin is blocked from binding to actin by this protein called tropomyosin. This is why muscle cells are not just constantly contracting, because the myosin physically cannot grab onto the actin unless we give it permission to. It can't grab onto the actin because the actin is blockaded by this protein called tropomyosin. There's another protein here. He's a regulatory protein called troponin. All right. We know, let's backtrack for a second. We know an action potential was induced in our muscle cell because of acetylcholine released by a motor neuron. The action potential travels along the muscle cell, dives down the T-tubules, next to the terminal cisternae of the sarcoplasmic reticulum, and it causes those cisternae to release calcium, and then calcium rushes into the sarcoplasm. In order for contraction to happen, we know myosin needs to grab onto actin, but actin is usually blocked by tropomyosin. Well, what exactly do you think calcium is going to do? If you said calcium is going to move this tropomyosin, you are 100% correct. Let's see what happens. So here is actin, here is myosin. The head wants to grab onto it, but it can't because it's blocked by tropomyosin. In the presence of calcium, here comes calcium. Calcium is going to bind to the regulatory protein troponin. So there's calcium binding to troponin. When that happens, the tropomyosin shifts. And now the actin is exposed. Exposed to whom? To myosin. Myosin grabs on. And notice how myosin's bent here now. Myosin has now pulled actin inward, and the muscle cell has contracted. And by the way, this is called a myosin crossbridge, where the myosin and actin have interacted like this. So, let's backtrack for a second, guys. Let's backtrack. Whoa, not four-track, backtrack. All right, it's my muscle cell right here. It's got an action potential. Dun, 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 because it got acetylcholine from a motor neuron. Action potential goes down a T-tubule. Neighboring sarcoplasmic reticulum, the terminal cisterna, releases calcium. Calcium then binds to the troponin on the thin filament. The troponin then, with the help of calcium, moves to tropomyosin. Actin is exposed. Myosin grabs onto actin, and myosin pulls actin inward, and the muscle cell is going to contract because the sarcomeres are contracting. Let's look at that interaction between myosin and actin a little more. So, starting out here, number one, it's a good place to start. Myosin has grabbed onto actin. It's able to do so because these little black circles here are the binding site, and they're exposed because tropomyosin is out of the way because troponin has bound to calcium. Now, once myosin has grabbed on, myosin is going to pull inward. It's called the power stroke, where myosin pulls actin inward. When it does that, it releases a phosphate. Notice that the myosin here has a ADP and a phosphate. What that means is that the myosin was in a high energy state. It had broken down ATP. And when it releases the phosphate, myosin then is able to pull actin inward. Then myosin releases the adenosine diphosphate, the ADP. Then myosin binds to a new ATP and lets go. And now, myosin can break down that ATP, get high energy again, grab on again, and pull inward again. Think about if you were like pulling on a rope, hand over hand over hand. Well, myosin can keep pulling on actin, pulling it in and in and in and in, and the sarcomere gets shorter and shorter and shorter and shorter. And can keep doing this as long as it has ATP and as long as calcium is there to keep the actin exposed. All right, wow. I know this is a ton. I'm like going kind of crazy with this stuff here. So I encourage you to pause for a minute. Try and write down all these steps. Okay? Try and write down all these steps. Try and organize it. Putting this stuff in proper sequence is a great thing to put on a test. So right now, try and do it. Write it out. Get your notebook out. Watch this over and over again until you've got a good order of these things occurring. Okay. Once contraction is done, well, the sarcoplasmic reticulum is going to steal the calcium back. There's a protein called the sarcoplasmic reticulum calcium ATPase, which uses ATP to kind of like vacuum the calcium back into the sarcoplasmic reticulum. Once that calcium starts to disappear, it's no longer going to be binding to troponin. So troponin and tropomyosin are no longer going to be in the same position. Myosin can no longer grab onto actin. So we don't have any more of these cross bridges. And then what happens is the muscle's going to return to its resting length. It does this because the connective tissue that got stretched because of contraction is going to recoil. Plus gravity is going to help as well. Flex your elbow right now. Contract your biceps, break, you got to do that. And then let go, and gravity will help you bring that muscle back to normal. So this is how we get back to normal after muscle contraction. We got to get rid of the calcium, which moves the tropomyosin, which prevents further interaction between myosin and actin, and we go back to the resting length. All right. Fantastic. Let's keep going. Good news is we're done with that discussion of kind of like the microscopic view of muscle contraction. We're going to move on to another big topic. You guys ready for another big topic? I hope so. All right, let me ask you a question. Earlier we said the contraction of a muscle cell was an all or nothing event. So every muscle cell contracts either all the way or not at all, okay? An individual muscle cell contracts all the way or not at all, all right? This kind of creates a dilemma. Think about holding your arm out, abducted, like you're making a T with your arms and your body, okay? Does it take a lot of strength just to hold your arm out? Not really, right? What muscle is holding your arm out in that abducted state? If you said deltoid, you are correct. All right, but now let's say instead of just holding your arm out, you were holding 10 pounds in that hand. It's going to make it harder to keep that arm out, isn't it? Definitely. You got to generate more force, more power, more tension, however you want to phrase it, you need more. But how are we able to have more if muscle fiber contraction is an all or none event? Well, there's a trick here. Every muscle fiber individually is all or nothing. But I can vary how many muscle fibers I contract. Maybe I contract 10, maybe I contract 20, maybe I contract 30. Depending on how many muscle cells in my deltoid actually contract will determine on how much tension I create. If I contract more muscle cells, I get more tension. All right? And to figure out how we do this, we have to introduce ourselves to a concept called the motor unit. A motor unit is a motor neuron. So for example, let's look at this purple guy here. A motor neuron like this purple guy, and all the muscle fibers it goes to, like these purple muscle fibers. So one motor neuron and all its muscle fibers is a motor unit. So if this motor neuron, the purple one again, sends an action potential, I am going to contract just the fibers that are purple here. I'm just going to contract that motor unit. If I want more tension, I'm going to send signals down this red motor neuron and contract another motor unit. If I contract the red and the purple motor units, I get more muscle fibers contracting and I get more tension. If I do all three, well, then I'm going to have even more tension. So even though a muscle cell is an all or none thing, its contraction is an all or none thing, I can vary the number of motor units I stimulate, and therefore I vary the amount of tension I develop. Motor units is proportional to tension. That's a proportional sign right there. Now, if you hold your arm out in that abducted position and you just keep it there for a minute, you'll switch from one motor unit to another, to another, to another. As one becomes fatigued, you'll switch to another, which is pretty cool. All right. Let's see. There we go. Just showing you this example here again. Here's my muscle right here, and these are muscle fibers in my muscle. And when I'm contracting none of them, none, I'm going to get no tension. If I start contracting more motor units, I'm going to start getting more tension. If I contract all my motor units, I am going to get my maximum contraction. Now, pretty cool fact, if you do not lift weights, then you are probably not very good at getting all your motor units to contract. Unless you move heavy things regularly, you're not going to be good at using lots of motor units. So if you started lifting weights today, which is really a great idea, if you don't lift weights, lift weights. But if you started, you are going to get stronger. In the first several weeks of working out, you will get stronger. Now here's where it gets crazy. You'll get stronger before your muscles get any bigger. And this sounds like it's defies common sense. But what's going to happen is in this initial beginner stage of weightlifting, of weight training, you get better at recruiting more motor units. So let's say you start out, you can only recruit 65% of your motor units in your pec major. But with a few weeks of training under your belt, now you can recruit 85% of your motor units. So even though the muscle itself hasn't gotten any bigger or any, stronger, but it can generate more tension because you're better able to use it. All right, fantastic. Okay. Do me a favor, name this muscle right here. No hints. You don't need a hint. Name that muscle. You know it. It's big. And there I just gave you a hint. So gluteus maximus. All right, name this muscle right here. Straight on the outside of the eye. It's the lateral rectus. Hopefully you said that. All right, which of these muscles, gluteus maximus or lateral rectus, needs finer movements, more delicate movements? It's the lateral rectus. So the lateral rectus will have smaller motor units than the gluteus maximus. The gluteus maximus will have larger motor units. So you'll generate tension in your gluteus maximus in bigger steps. You can generate tension in your lateral rectus in smaller steps and leading you to finer movements. So the point I'm trying to make here is motor units vary in size. Different muscles have different sized motor units. Smaller motor units lead us to finer movements. So your eyeballs, your fingers, places where you need fine, delicate movements, you need you need small motor units. All right. Cool. New topic, guys. New topic. Related, but still new. When we get a muscle fiber to contract, just to have one contraction and then relax, that's called a twitch. So if I stimulate my muscle cell at time 0, there's a little period where nothing happens yet as far as generating tension. Then there's a period where tension is generated, it's a period of contraction, and a period where tension falls, it's a period of relaxation. And the whole combo of these three, the relaxation, the contraction, and the latent period where no tension is developed yet, that's called a muscle twitch. Now you're probably wondering, what's going on in the latent period? Well, remember, when we stimulate a muscle cell with acetylcholine, we have to generate an action potential. The action potential has to go down the T-tubules, has to get the sarcoplasmic reticulum's terminal cisternae to release calcium, which then has to bind to troponin, which has to move tropomyosin. Then myosin can grab and pull on actin, and we can start to contract and generate tension. And so all that stuff in the beginning there was during the latent period. So this is a muscle twitch graph right here. And, if I can get to the next slide, I got another muscle twitch graph here as well. The time scale changed. That's why it looks a little different. So the latent period is hard to see. Contraction, relaxation. Stimulate a muscle fiber, we get a twitch like this. But you know what we can do? We can stimulate it more than once. This is really cool. Let me go back for a second here. Look at where we got. Look at the amount of tension we got right here. That's how much tension we got, like right there, right on the E-ish, okay? And we stimulated the muscle fiber again right here. It started to contracted, generated tension, all the way up to the E. And it started to relax. But we didn't let it relax all the way. We hit it again. We stimulated it again. And it started contracting again. And it went up even higher. Look at that. It went up even higher. In that second stimulus, we got it to contract even more. Then it started to relax. And then boom, we hit it again. And boom, we hit it again. And we keep generating more and more tension until we get to the maximum stage of tension. The maximum stage of tension. The maximum stage of tension is called tetanus. All right? The max contraction is tetanus. All right? I know you might be thinking about, well, what about tetanus, like the tetanus shot? And we'll talk about that in a second. But tetanus refers to this maximum state of contraction. Okay? This is called unfused tetanus because we had these periods of relaxation in between. So if we keep stimulating a muscle cell over and over again, we are going to get to tetanus. And this is unfused or incomplete tetanus. Now, if I bring those stimuli closer together, if I'm just releasing acetylcholine, boom, 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 on that muscle fiber, I'm never giving it a chance to relax. I'm getting the tension to go all the way up to tetanus, and it stays there. This is called fused or complete tetanus. Okay? So, if we repeatedly stimulate a muscle fiber, we can get it to generate more tension. Repeatedly stimulate it, we can get it to generate more tension. All right. Fantastic. Let's talk about that other meaning of the word tetanus, the other idea, like a tetanus shot. Well, there's this bacterium, we see these little purple guys right here, called Clostridium tetani. If you've taken microbiology, you'd understand that where it says it's a gram-positive spore-bearing rod. You'd get all that. This bacterium, you can also often find it on like classically on like a rusty nail or rusty barbed wire. And so if you cut yourself on this barbed wire, you can get this bacterium to go into that wound. The bacterium is going to eventually get to your spinal cord and brainstem by way of neurons and your bloodstream. And motor neurons in the spinal cord and the brainstem become hyperactive. And what it's going to do is it's going to cause the muscle fibers to start to contract, to have this uncontrolled contraction. We can see this guy here. You can see his platysma is contracting. He's got all these facial muscles. He's pulling his teeth are clenched. This is called lockjaw. Think about what muscles would cause you to clench your teeth. The masseter, the the temporalis. We can see he's contracting a lot of muscle here. His arms are contracting. He's in this spasm of contraction here. It can happen in animals. It's a horse experiencing tetanus. It can happen in babies, especially if there's like non-sterile delivery, like non-sterile cutting of the umbilical cord. So this is the relation between tetanus, the bacterium. A tetanus shot, of course, lets your immune system protect against this bacterium and its toxins. And if you haven't had a tetanus shot in a long time, it might be a good idea to get one. So there's a relationship here between tetanus, the bacterium, and tetanus, the idea of complete muscle contraction. All right. Wow. That took longer than I wanted it to. You know what, guys? I think I'm going to break this up into 3 videos in total on muscle, so I'm going to quit here. We're going to do one more video on muscle for the next time. So take a little break. I'll see you later.